Hey everyone, welcome to the Wise Tutor YouTube channel. Today's video is video number 8 about enzymes. Enzymes are proteins that help catalyze or speed up the rate of chemical reactions, and they do that by lowering the activation energy, which is the energy required for the reaction to occur or start. And so here you've got over the progress of a reaction, you're plotting the activation energy. And you see a solid curve here, starting from the reactants and ending in products, where you see a very high activation energy without the enzyme. And then you see the dotted line, which is a lot lower in activation energy. And that is because of the presence of an enzyme in that reaction. So that is the main sense of the main function of an enzyme. A reaction that would take a lot longer time could be sped up uh, uh, with the help of an enzyme. And so here are a couple examples. Amylase is a digestive enzyme that digests carbs. Pepsin is also a digestive enzyme that digests proteins in the stomach. And polymerase is an enzyme useful for DNA and RNA synthesis. And there are multiple types of polymerases that we'll talk about later on. And now we're looking at factors that impact the enzyme structure. And whenever the shape or structure of an enzyme changes, so does the function. So here we have an enzyme before a stage known as denaturation. Now, after one of these factors takes place, we have the enzyme after denaturation. So you see that the entire shape of the enzyme changes, and thus uh, enzyme this enzyme here no longer has the function that the previous enzyme had because its shape has changed dramatically. So those two factors are very high temperatures and extreme pH changes. And so very high temperatures, as you keep adding that thermal energy due to the high temperatures, it starts breaking the hydrogen bonds and other interactions within the protein. Remember, because enzymes are proteins. So the hotter or the more thermal energy is input in the protein, you're going to start breaking those hydrogen bonds and other interactions. And this will change the shape and denature the enzyme of the enzyme. Here you've got extreme pH changes, and that really affects the functional groups on the amino acids of the enzyme. So the enzyme is just like any normal protein. It's made of a sequence of amino acids held together by peptide bonds. And you could go rewatch the the proteins video, and that'll help you out. But anyway, you see the alteration of the functional groups chemically, and that is really what denatures and ends up denaturing the enzyme and rendering it useless for future functionality. And so here's the picture that I've already explained. You see here the enzyme completely changing shape. Even the active site of the enzyme here is no longer the same shape that it used to be. And thus, when a substrate tries to come in and bind to the active site, you see that it's unable to because it's shaped like this. The enzyme is shaped to fit this. The substrate is shaped to fit this shape of the enzyme. Unfortunately, when the enzyme shape changes like this, the substrate itself cannot change to kind of bind in there. So that is how it changes the function. So here we see the formation of an enzyme substrate complex where there's the enzyme and the active site and a substrate binds. When it binds, you get the formation of the complex. And then eventually, soon afterwards, you get the formation of products from the substrate and the enzyme. And this is done by several chemical reactions. The products could then go wherever they need to trigger a reaction, or maybe that's the reaction that is maybe pumping out things in or out of a cell, or even if it remains in the cell. And we'll talk about some examples of specific enzymes and what happens to the products after they are created. But you could see how this is potentially problematic, can get problematic. You see, if we make too many products and we want to stop whatever process, how do we do that? How do we do that in a cell or in the human body, or actually in any organism's body? Well, the enzymes do need a robust method of regulation, and they do this by several different types of inhibitors that can slow down or stop enzymatic activity completely. And this is really important. We'll talk about three types today. The first one is the competitive inhibition, and that's when an inhibitor competes against the substrate for the active site. And it also make sure to know that it can be overcome with more substrates, and we'll explain both of that using this diagram here. You've got the enzyme and the active site, and you've got a substrate versus a competitive inhibitor. If the competitive inhibitor is present and it can fit in the active site, which that is the definition, it will fit in the active site, and it will compete with the substrate for the active site. When it binds, the substrate doesn't have anywhere to bind itself, and so you don't get the reaction, or you don't get the products that are usually created by the enzyme complex. And there it is shown right here. Now, how can it be overcome with more substrate? Well, the more substrate surrounding the enzyme or around this area, the less of a chance that one or a couple competitive inhibitors will find their way to the active site. The more substrates, they're kind of taking an army versus a couple competitive inhibitors. So there's a higher chance for a substrate to bind to the active site, and that's really how the body that's really the method that the body starts inhibiting enzyme complexes from forming, and by increasing the substrate, you could kind of reverse that itself. So here we have substrate concentration plotted against the rate of reaction, and you see a pretty normal curve when there's no inhibitor present. But when there is a competitive inhibitor present, you see a lower curve. We need a much higher substrate concentration to achieve the same rate of reaction, and that is because that competitive inhibitor needs more competition from the substrate side so that it doesn't end up binding and stopping the reaction itself. And that's really important. Now we also have another method of non-competitive inhibition. And this type of inhibition doesn't attach on the enzyme's active site. So say this is the active site here. It doesn't. It instead goes for a different site called the allosteric site. This is a site where the inhibitor binds and ultimately triggers a change in the shape of the enzyme and the original active site. So here is the active site, and here is the allosteric site. When the inhibitor, which is this blue circle, binds to the allosteric site, you see that the shape of the active site itself changes. So the substrate that usually could have fit in the original active site can no longer fit. And so this does not involve the inhibitor actually going into the active site. Instead, it is a different site called the allosteric site again. And so as you may or can be able to guess, this is not really fixed by an increase in substrate, because if the substrate shape is if this active site shape is changing itself, you see that you're not able to fit any of these substrates, no matter the number. And so that's really important distinction between competitive and non-competitive inhibition. The third type is called allosteric regulation, and there's two types. There's allosteric inhibition and there's allosteric activation. So here you've got an enzyme for the inhibition part. Let's talk about the inhibition side first. And you've got an active site on it. And then you've got a similar allosteric site as we were talking before. When you put the inhibitor, the shape of the active site itself changes. So the substrate is unable to fit it. And so this is really similar to the non-competitive inhibition. All right. But the allosteric activation actually works the opposite way. At first, you have an altered active site, all right? And the substrate cannot fit in the altered active site. But then you put in an activator molecule, all right, into an allosteric site. What that serves to do is change the shape of the active site to make it functional for the substrate to bind there. And so you see a fundamental change in shape of the enzyme, and that allows the substrate to bind and for products to be made, or the process or chemical reaction to be catalyzed. And so it's important to realize that whether if it's an inhibitor or an activator, in this case, these molecules are called effector molecules. They affect somehow the trajectory of the enzyme regulation. And so that's really important for the allosteric regulation. Now, reaction rate. So reaction rate is affected by a couple or three actually factors. The first one being pH. In order for the reaction rate to be the highest it can be, the pH must be optimal for the specific enzyme. The second one is temperature. The high temperatures are usually that are still optimal for the enzyme, usually increase the reaction rate by increasing the frequency of molecular collisions. And we'll explain these down below, no worries. Finally, the third factor is substrate concentration. The more substrate, the faster the rate of reaction, until this thing called Vmax is reached. Vmax is the maximum rate of reaction when the enzymes themselves no longer have an open active site, which means that a substrate is already bound to the maximum amount of enzymes present in the system. And so after that, you can't really go above that Vmax, okay? And so that rate of reaction slows down at that point at Vmax. Now, when we're talking about pH and temperature, you see a similar curve to this. Here, this is specific to temperature, but you could imagine a pH scale being on here if you want to. And I'll go through each of these. The temperature, as you see, as temperature increases, at least for the first half, you see an optimal temperature where the reaction rate is the highest it will ever be for that specific enzyme. So for this case, the optimum temperature ends up being roughly less than 45 degrees Celsius. So we'll call it 43 or something. I think from down there, 41, 42, or 43 degrees Celsius. And that is the optimal temperature. That means any temperature above it, you'll start falling and the reaction rate will start decreasing. Any temperature below it, the same thing will happen. It'll start falling, the reaction rate will at least, and you'll see lower reaction rate as temperature decreases and lower reaction rate as temperature increases. There's usually just an optimal temperature or a set of temperatures that really maximize the rate of reaction. The same thing happens when pH is on this x-axis. You see a pH and you've seen an optimal pH. Say in this case, this was from acidic to basic from 0 to 14, and say right here turned out to be like 9, so a bit basic. And so say that's the optimal pH. Anything above that optimal pH will start denaturing the enzyme. So that's not good for it, and that'll decrease the rate of reaction. Anything below, again, will start denaturing the enzyme, and you'll see lower rates of reactions. Now, the final one is the substrate concentration versus the reaction velocity of reaction speed, or rate of reaction. So you see this curved image, curved graph, until we reach Vmax, which at that time, the reaction rate becomes constant, all right? And you see that at the Vmax, you see a greater amount of substrates is needed, but then once each substrate is bound to the max number of enzymes in the system, there's no more enzymes for the substrates to bind. So no matter how much you increase the substrate, you won't be increasing the reaction rate because there's no more enzymes to bind to the substrate. And so that's really important as a kind of thing to note, that the reaction rate can actually be influenced by these three factors. And it's also important to know how they are influenced.